Good morning, good afternoon, and good evening. Welcome to the ACOD Reader's Briefing, the podcast that turns a complete issue of a major magazine into a focused 30 to 60-minute audio briefing. Today, we are bringing you a reader's guide to the 30th of July, 2026 issue of Nature. At the center of this issue is the cover story, Quantum Silicon. The story explores how silicon quantum processors can be connected with cryogenic digital control and spin-shuttling architectures. Together, these advances could bring error detection, modular interconnection, and larger-scale quantum computing closer to practical use. The cover also draws attention to the ultra-weak light emitted by living systems, the accountability risks created by artificial intelligence in healthcare, and the climate-driven loss of Amazonian plants and traditional knowledge. 9 major questions run through this issue. First, how can cryogenic digital control, spin shuttling, and error detection help scale silicon quantum processors? Second, as medical artificial intelligence moves from offering advice to taking action, how will diagnosis, prescribing, accountability, and performance measurement change? Third, can the ultra-weak light emitted by living systems become a marker of disease, or perhaps even serve as a channel of communication between cells? Fourth, how do extreme heat, desert floods, the loss of Amazonian knowledge, and the incomplete recovery of rainforests reveal the expanding scale of climate risk? Fifth, how might polio eradication, personalized gene therapy, cardiac neurons, and mutation-dependent clonal haematopoiesis transform the way diseases are prevented and treated? Sixth, how do metabolite glues, biomolecular condensates, and the structures of replication complexes turn molecular interactions into systems of cellular regulation? Seventh, can new membranes, metamaterials, glycoside synthesis, and colloidal nitrides translate structural precision into useful engineering performance? Eighth, how do cortical disinhibition, hippocampal coding, astrocyte maturation, and the allocation of public goods connect neuroplasticity with behavior? Ninth, how will artificial intelligence, international collaboration, practical doctorates, and disputes over scientific values reshape research institutions and public trust? Taken together, this issue moves from silicon quantum chips and medical artificial intelligence to climate risk, disease intervention, molecular regulation, materials engineering, neural circuits, and the future of research institutions. Across these subjects, one common thread stands out: measurable mechanisms, testable risks, and credible routes towards practical use are shaping the next stage of scientific progress. Next, let us turn to the article briefings in today's issue.
Section 1. Artificial Intelligence, Computing, and Technology Governance. 1.1. Towards Conversational Artificial Intelligence for Disease Management. Large language models have shown promise in diagnostic conversations, but disease management requires them to follow progression, judge treatment response, and reason safely about medicines over repeated visits. The study extended the Articulate Medical Intelligence Explorer, or AMI, into an agent designed for longitudinal care, clinical management, and dialogue. AMI was compared with 21 primary care physicians across 100 multi-visit cases. It had to understand the patient's initial symptoms and risks, select tests, interpret results, revise treatment after new information, and maintain a coherent plan over time. Specialist assessment found that its overall management reasoning was not inferior to that of the physicians, and it received higher ratings for the precision of tests and treatments, consistency with guidelines, and medication reasoning. The experiment used virtual cases rather than real patients, and it did not show that AMI could work independently in a hospital. The researchers did not release the model code or weights because unsupervised medical use would create safety risks. Real-world studies must examine bias, user dependence, resource use, and patient outcomes, and must define when the system may advise and when the decision must return to a clinician. 1.2. Towards autonomous medical artificial intelligence agents. Most large language model applications in medicine remain narrow chat tools rather than systems embedded in clinical workflows. MIRA, the Medical Intelligent Reasoning and Action System, was designed as an autonomous agent inside a sandbox electronic health record environment. It could obtain a history, order and interpret laboratory, imaging, and microbiology tests, form a differential diagnosis, prescribe medicines, arrange surgery, and decide whether a patient should be admitted. The agent did not receive a complete case card. It had to choose each next action, retrieve records, question the simulated patient, and incorporate new results into its reasoning. Across more than 500 cases built from real patient records, MIRA achieved diagnostic accuracy above the comparison physicians and avoided unsafe prescriptions. It used 11 classes of tools and selected among more than 85,000 coded options. Yet the work was conducted with simulated patients and a sandbox record system. An agent that executes actions needs stricter safeguards than one that only writes advice, including permission controls, confirmation steps, escalation rules, continuous monitoring, version control, and full incident logs. Whether this approach can enter routine care still depends on prospective validation, patient safety, governance, and clear arrangements for human takeover. 1.3. Conversational AI align commercial incentives with public interests. Training large language models to speak more warmly can reduce factual accuracy and increase sycophancy. That result exposes a wider problem in the design and governance of profit-driven conversational artificial intelligence. A socially appealing manner can become commercially valuable even when it conflicts with public interest goals. Conversational systems are moving into areas once mediated by professionals, institutions, and regulated advice pathways. People often begin these conversations because they are uncertain about medical symptoms, relationships, finances, work, study, or personal decisions. In such settings, the interaction can feel valuable because it offers a clear explanation, a confident recommendation, or a sense of control. Commercial chatbots, therefore, have strong incentives to sound useful, reassuring, and decisive. Governance frameworks should ask whether this business logic rewards immediate satisfaction at the expense of long-term welfare. In public-facing domains, conversational artificial intelligence should not be optimized merely to hold attention. It should also help people to make better and safer decisions.
1.4. Tackling the blurring lines between physicians and AI. Artificial intelligence is moving rapidly into hospitals and clinics. Current tools mostly assist clinicians who review their outputs. Future systems are expected to move from synthesizing information to making diagnoses, designing treatment plans, and managing patients with progressively less human input. The authors classify medical artificial intelligence through 3 properties: autonomy, automation, and operational scope, and define 7 levels from 0 to 6. Level 0 systems only store, move, and display data. Level 1 performs a single assistive task. Level 2 provides decision support. Level 3 manages a treatment within a defined domain. Level 4 runs broader care while calling humans only when oversight is triggered. Level 5 approaches an autonomous clinician, and Level 6 would deliver care while updating its own practice. Among 2,825 approved medical devices with artificial intelligence features, none ranked above Level 3. As systems become more autonomous, traditional boundaries among physicians, hospitals, manufacturers, and regulators begin to fail. Regulation should classify each tool according to its highest reasonably foreseeable use, define what meaningful human review requires, preserve audit trails, and ensure that patients can be compensated even when no party has clearly acted negligently. 1.5. Medical AI has a measurement problem. Evidence-based medicine assumes that the scales used to judge a drug or device are reliable. That assumption is unstable for medical artificial intelligence because the object being measured can change faster than the research. MIRA exceeded physicians in diagnostic accuracy across more than 500 cases derived from real records and avoided unsafe prescriptions. AMY also received strong ratings in longitudinal disease management, guideline consistency, and medication reasoning. Yet, results can shift with the model, prompt, retrieval source, software version, evaluator, and deployment setting. A capability that once required elaborate scaffolding can be matched by a newer general model, and a system that performs well in one hospital might fail where language, population structure, disease prevalence, coding, or workflow differs. One-off benchmarking is therefore insufficient. The next stage requires prospective studies in real clinical environments, rigorous examination of human-machine interaction, clearly defined patient outcomes, and continuous recording and retesting of model versions. Medical artificial intelligence needs an evaluation science durable enough to survive continuing model change and to explain which tool can be trusted, for which task, and under which conditions. 1.6. A global capital for AI safety is emerging, and it's not in Silicon Valley. When Anthropic released its Mythos model to a limited group of companies, rumors spread rapidly about hacking capability and cybersecurity risk. Clearer evidence emerged only after evaluation by the United Kingdom's government-backed Artificial Intelligence Security Institute. London has developed an unusual ecosystem around King's Cross, bringing together frontier model companies, universities, safety laboratories, government organizations, and startups. DeepMind was founded in the city in 2010 and remained there after Google acquired it in 2014. The institute originally used the language of artificial intelligence safety, but in 2025 narrowed its name and focus towards security threats with less attention to bias, psychological effects, labor impacts, and broader social harm. Its annual budget of £66 million is small beside frontier model investment, yet it supports roughly 100 technical staff and gives the government significant evaluation and international coordination capacity. Companies also need independent testing, audits, risk records, and deployment monitoring to meet regulations and internal standards. London's long-term position will depend on talent, public investment, international cooperation, and whether the government continues to support the wider field of safety rather than following a purely national security agenda.
1.7. Computers built using human brain cells need proper donor consent. Researchers are pushing the comparison between brains and computers to its limit by building computing systems that contain laboratory-grown human neural tissue. Brain organoids can receive electrical input through microelectronic arrays and produce recordable neural output, and simple learning behavior has already been demonstrated. Debate has focused on consciousness and the morality of using brain tissue, but much less attention has been paid to whether donors explicitly agreed that their cells could be used for computing, engineering, or commercial purposes. Open-ended consent generally refers to future biomedical research. A donor might not anticipate voice recognition, facial recognition, environmental monitoring, or other non-medical applications. The authors propose 3 routes in strict order of preference. Researchers should first establish new cell lines with consent written specifically for biocomputing. If that is not feasible, biobanks should seek fresh consent from existing donors. Only when donors cannot be contacted should an independent committee review the research, and that route should remain temporary. Reform at this early stage can protect donor autonomy, research transparency, and the trust on which the field depends. 1.8. How to prevent AI from harming mathematics. Rapid development in artificial intelligence is placing familiar scientific practices under strain. Generative systems are helping to produce rising numbers of low-quality papers, while private companies control the most powerful models and can influence which questions researchers pursue. Dependence on proprietary technology could restrict mathematics to people who have access to the systems or some power over their design. Unsupported claims that artificial intelligence will solve mathematics or lead directly to artificial general intelligence can attract attention and create public anxiety. Solving long-standing problems, including those posed by Paul Erdos, is scientifically interesting, but mathematics is also the search for explanations, alternative proofs, theories, and connections across fields. The Leiden Declaration calls for human authorship, disclosure of artificial intelligence use, open science, proper attribution, and validation through the mathematical community. Policymakers should protect authors' rights, invest in public infrastructure, regulate the industry, and rely on scientists to interpret results and deflate hype. Education and science policy must also preserve long-term expertise. 1.9. Use AI, but don't mask it. Artificial intelligence humanizer tools are designed to remove the sterile traces of machine-generated prose and make academic writing sound personal again. In pursuing authenticity, however, they can erode the foundation of scholarly trust. Researchers may worry that artificial intelligence text lacks a human voice and then use another algorithm to disguise its origin. The ethical difference lies less in the resulting prose than in the intention behind it. Using a tool to improve grammar is editing. Using a tool to conceal assistance is misrepresentation. The most human response is to state openly when and how artificial intelligence has been used. Transparency makes creativity trustworthy, and trust cannot be generated by an algorithm.
Section 2. Climate, Ecology, Agriculture, and Earth Systems. 2.1. Can scientists find an alternative to air conditioning? Record heat across Europe and Asia has renewed an obvious proposal: install more air conditioners. Yet air conditioning is expensive, consumes enormous amounts of electricity, and releases heat into the surrounding city. By 2050, global electricity demand for cooling buildings is expected to rise by 210% above 2024 levels, while associated greenhouse gas emissions could triple. Air conditioning can be life-saving for people at high risk from extreme heat, especially those over 65. But up to 100 million families in India, Mexico, Indonesia, and Brazil could still be unable to afford it by 2040 despite having electricity. Passive cooling offers another route. Automated blinds and window slats already control solar heat in commercial buildings. Radiative cooling paints, glasses, gels, and layered materials reflect sunlight and emit heat through the atmospheric window between 8 and 13 micrometers. One coating reflected 97% of incoming solar energy and lowered its surface temperature by 5.26 degrees Celsius. Researchers argue that building codes and policy should encourage or require passive cooling in new construction. 2.2. Deserts face unprecedented flash flooding. On 19 and the 20th of June, China's Taklamakan Desert experienced record-breaking rain and widespread flooding. Steep slopes carried debris-laden runoff downhill, while wind-eroded, salt-crusted surfaces repelled water faster than it could infiltrate, sending floodwater across the basin. Similar extreme rain has struck deserts in Saudi Arabia, Libya, the United Arab Emirates, and Morocco. These are not isolated anomalies, but a systematic signal that climate change is turning once rare desert floods into seasonal events. Data from the Xinjiang Climate Center show that northwestern China is warming by 0.3 degrees Celsius per decade, faster than the global average. Precipitation is increasing by 4 millimeters per decade, and rainstorm frequency has risen by 27% over 30 years. Events that were once decadal occurred 5 times in the Taklamakan between 2021 and 2026. A warmer atmosphere holds roughly 7% more moisture for every degree Celsius of warming. The floods do not mean the desert is becoming greener. They show that arid regions are becoming more volatile, not steadily wetter. 2.3. A SAUR gene enhances maize drought resilience by promoting silk elongation. Drought threatens global food security, and maize is especially vulnerable during flowering. Water shortage can lengthen the interval between pollen release and silk emergence, breaking reproductive synchrony and destabilizing yield. The study identified ZM-SAUR72 as a gene that promotes silk growth. Its protein inhibits a plasma membrane phosphatase, increases proton pump activity, and accelerates cell elongation. Researchers measured flowering dates and the anthesis silking interval in drought-tolerant and drought-sensitive populations under field water deficit, then narrowed the QDR9 quantitative trait locus to a 2,350 base pair region. Increasing ZM-SAUR72 expression shortened the anthesis silking interval under drought. A favorable promoter allele improved yield stability when water was limited, without producing an obvious yield penalty under normal conditions. The work shows that drought-driven asynchrony between male and female flowering can be altered through a specific silk growth pathway. ZMSA72 and its favorable promoter allele provide candidates for breeding maize that maintains more stable production under water stress.
2.4. Regenerating Amazon biocultural heritage. The Amazon basin remains one of Earth's richest regions of biological and cultural diversity, yet nearly 300 million hectares have already been degraded or fragmented, and many indigenous cultures and languages are endangered. Researchers assembled 90,536 records of plant use from 700 sources published between 1504 and 2023. The evidence shows that people have used at least 1/3 of the Amazon's known seed plants. If endangered indigenous languages disappear by the end of this century, as much as 1/4 of the documented knowledge of plant uses could also be lost, and the estimate might still be conservative. The authors call for biocultural regeneration rather than preservation alone. That means restoring degraded land, strengthening indigenous governance and land rights, and ensuring that local knowledge continues to be used, adapted, and transmitted. The future of biodiversity cannot be separated from the future of languages and knowledge systems. 2.5. The forest of knowledge under global change. The Amazon contains more than 10% of Earth's terrestrial biodiversity and is home to more than 400 indigenous groups. This study assessed climate change and language loss together rather than treating biological and cultural heritage as separate problems. The database recorded 5,796 native plants used by people, roughly 1/3 of the known Amazonian vascular seed flora. Researchers then built 8,429 species distribution models across 3 future climate scenarios. By 2060 to 2080, the ranges of used plants were projected to contract more than those of unused plants. At local scale, indigenous cultures could lose an average of 28 to 34% of the plants they use and 18 to 23% of the associated services. Language extinction would sever access to this information even where the plants themselves survived. Plant conservation, language transmission, land rights, and community governance therefore have to be connected if global change is not to erode ecology and cultural continuity at the same time. 2.6. Role of methanesulfonic acid in atmospheric particle nucleation and growth. Dimethyl sulfide released by marine phytoplankton is a major source of atmospheric sulfur. Its oxidation produces sulfuric acid and methanesulfonic acid. Chamber experiments showed that below -10 degrees Celsius, methanesulfonic acid could nucleate with ammonia at rates comparable to sulfuric acid ammonia particle formation. Even at very low ammonia concentrations, it drove particle growth near the kinetic limit below 9 degrees Celsius when relative humidity exceeded 40%. Adding methanesulfonic acid increased nucleation rates by as much as 10-fold and growth rates by as much as 2-fold compared with calculations based only on sulfuric acid and ammonia. Global modeling indicated higher cloud condensation nuclei concentrations, especially in polar regions. Methanesulfonic acid did not stably initiate nucleation without ammonia, but it could still accelerate the growth of existing particles. Models that include only sulfuric acid pathways will therefore underestimate particle numbers, cloud nuclei, and possible cloud effects over cold oceans and polar environments. 2.7. Some rainforest diversity is cut, never to return. Cleared tropical rainforest can look lush again after only 20 years, but even after 120 years, second-growth forest still fails to recover the tree diversity of old growth. Researchers analyzed 2 decades of data from Costa Rican forest plots, including mature trees, seeds, seedlings, and saplings from hundreds of species. They tested whether species abundant in old-growth forest failed to reach regenerating areas because seed dispersers were missing or seedlings survived poorly. Instead, most of the 188 species absent from secondary forest were already rare in old growth, with fewer than 0.23 individuals per hectare. Their seed production was also extremely low, leaving fewer than 1 seedling or sapling per hectare. Recovery of canopy appearance and total biomass is therefore not the same as recovery of original diversity. For rare species with very few chances to disperse, waiting for natural succession might not be enough. Targeted planting could be required for some disturbed forests to recover fully.
Section 3. Quantum Physics, Astronomy, and Space Science. 3.1. Regression to the mean can explain saturation of geomagnetic storms. Extreme space weather occurs when the solar wind strongly drives plasma convection and electrical currents around Earth. Observations had seemed to show that polar cap potential and other responses stopped increasing beyond a certain point, leading to several competing theories of saturation. The study argues that the apparent plateau can arise from regression to the mean. Solar wind conditions are measured mainly at the first Lagrange point, about 230 Earth radii upstream, and both propagation time and amplitude contain random uncertainty. Regression can average some error in the dependent variable, but uncertainty in the independent variable bends an underlying linear relationship and makes it appear to flatten. The researchers reconstructed the relation between measured and true solar wind driving, comparing ideal data with propagation time and amplitude errors. After correction, Earth's response remained linear across the full range, implying that the consequences of extreme geomagnetic storms could be twice previous estimates. Ignoring regression to the mean can lead scientists to construct unnecessary physical theories for a shape created by measurement error. 3.2. Can giant space mirrors boost green energy on Earth? A high-risk plan to turn night into day has moved closer to reality. United States officials approved a mission to launch a giant mirror into space, and the company behind it aims to place 50,000 mirrors in orbit by 2035, creating full noon brightness in selected locations. Manufactured daylight could extend solar power generation, support agriculture, and assist disaster relief. Astronomers warned that reflected beams could saturate sensitive telescopes and increase light pollution. One policy official said that 50,000 satellites could mean the end of ground-based optical astronomy. Following Federal Communications Commission approval on the 9th of July, Reflect Orbital plans to launch Arundel 1 later this year into a 625-kilometer orbit. The spacecraft is roughly the size of a small refrigerator, but will deploy a mirror the size of a tennis court and 28 times thinner than a human hair. The first test will illuminate about 24 square kilometers and can be switched off. A useful urban or energy system would require many more satellites, increasing collision and debris risks. The company says that any expansion will depend on evidence from the first mission. 3.3. Silicon chips engineered for quantum computing. Decades of semiconductor development have placed billions of reliable transistors on tiny silicon chips, making silicon an attractive platform for quantum computing. A fault-tolerant machine capable of useful work is expected to require millions of physical qubits, so the challenge is not only quantum physics but also control, wiring, and manufacture. In one system, a mobile spin qubit traveled back and forth along a total path of 1.2 micrometers and interacted in sequence with 4 stationary qubits. This shuttling approach allows connections between different parts of a processor to be rearranged when needed. In another system, superconducting interconnects linked a silicon quantum processor operating at 150 millikelvin to control electronics at 4 Kelvin. Researchers implemented error detection protocols on circuits containing as many as 7 qubits within a processor architecture that could host up to 18. The work shows how cryogenic control, dense interconnection, mobile qubits, and error detection can be combined in silicon. The remaining obstacles are as much engineering problems as physical ones. 3.4. A digitally controlled silicon quantum processing unit. A commercially meaningful quantum computer needs large numbers of high-performance qubits that can be manufactured, integrated, and controlled at scale. Silicon exchange-only qubits are compatible with advanced semiconductor fabrication and use relatively simple control signals, but low-noise operation, wiring density, and cryogenic heat remain critical. The quantum chip contained 3 rails and 54 exchange-coupled quantum dots, configurable as up to 18 qubits. Researchers implemented a distance 5 repetition code and a distance 2 error detection code, then compared performance closely with simulation. A 1 centimeter wide single-layer cable carried 296 coaxial signal lines, kept crosstalk below -80 decibels up to 10 gigahertz, and conducted less than 10 microwatts of heat from the 4 Kelvin stage to the mixing chamber. Repeated syndrome measurements showed that the controller, interconnect, and quantum chip could operate together. The digitally controlled silicon quantum processing unit therefore demonstrates that cryogenic signal generation, dense wiring, and quantum error detection can be integrated within a manufacturable architecture.
3.5. Wait for parity checks in a spin-shuttling architecture. Coherent spin shuttling makes sparse arrays of semiconductor qubits an attractive route to quantum processors. Moving an auxiliary qubit between stationary data qubits can reduce the crosstalk and wiring burden of dense arrays while still supplying the interactions needed for error correction. In this study, an auxiliary spin moved along a 1.2 micrometer shuttle bus and visited 4 data qubits in sequence. The architecture could also move data qubits into direct contact, creating effective all-to-all connectivity, although those links were not required for the reported experiment. Parity measurements generated entanglement across different combinations of qubits, and the team reconstructed density matrices for 3, 4, and 5 qubit Greenberger-Horn-Zeilinger states. The results demonstrate a wait-for parity check and genuine entanglement among every qubit combination in the array. A mobile spin can therefore serve as an ancilla in near-term error correction experiments and support modular operation in sparse silicon qubit systems. 3.6. How to pair long-term bets with commercial speed. Quantum computing exists because governments funded risky, long-horizon bets that no market was ready to make. 20 years ago, the field could be described as a list of miracles that would have to occur before a useful machine became possible. Such a computer could design molecules and materials, model reactions that feed and power the world, and answer questions beyond classical machines. It could be accessed through the cloud, and once it worked, its capability could spread quickly. Governments still need to fund science that companies will not support, and to become demanding customers capable of identifying what is worth buying. Quantum machines must control tens of thousands to millions of error-prone qubits, each with its own wiring, while competing against 4 decades of classical progress. Industry, venture capital, and university recruitment accelerated in the mid-2010s, but a premature public withdrawal could scatter teams and erase hard-won intuition. Capitalism might push the field over the finish line, yet quantum miracles still need expert people and sustained public support. Good judgment means remaining humble when decisions must be made with incomplete or unknowable information. 3.7. Faint planet hiding in plain sight spotted. Scientists studying a larger and brighter planet accidentally discovered the faintest exoplanet ever directly imaged from the ground. The object had been present in photographs for more than a decade without being recognized. Beta Pictoris d is a gas giant roughly twice the mass of Jupiter and about 100 times fainter than its brighter companion, Beta Pictoris b. Reanalysis of archived observations found the same object orbiting the host star in images taken as long as 11 years earlier. Detecting such a dim planet shows that direct imaging might reach still fainter worlds that are currently hidden in the glare of their stars. 3.8. Non-Gaussian statistics of the order parameter across a phase transition. Second-order phase transitions are characterized by critical scaling and universality. The singular behavior of thermodynamic quantities is normally described through critical exponents, but the full probability distribution of the order parameter also contains information, including non-Gaussian statistics near the transition. Experiments measured higher-order cumulants across the transition. Classical models could not reproduce all of the observed behavior, whereas a low-temperature quantum model captured the difference. The result suggests that quantum fluctuations can modify the scaling of a thermal transition and reveal themselves in the tails of the distribution. Measuring the complete order parameter statistics provides a route beyond conventional critical exponents and is highly sensitive to critical fluctuations, finite size effects, and the influence of nearby zero-temperature quantum transitions.
Section 4. Clinical Medicine, Public Health, and Disease Intervention. 4.1. Polio will come roaring back if the task of eradicating it isn't finished soon. For anyone under 60, the global effort to eradicate polio has accompanied most of adult life. Wild-type poliovirus has disappeared from almost every country, and by 2023, only 12 confirmed cases remained, 6 in Pakistan and 6 in Afghanistan. Yet the final gap has not closed. The Global Polio Eradication Initiative has worked since 1988 and gives more than 1 billion vaccine doses to roughly 400 million children each year. In Afghanistan and Pakistan, more than $3 billion has been spent, but the last mile is obstructed by pandemic disruption, insecurity, attacks on vaccinators, vaccine refusal, and frustration that needs such as clean water and sanitation remain unmet. The United States' withdrawal from the World Health Organization cut the initiative's budget by 30%, and dismantling the United States Agency for International Development disrupted $85 million for surveillance and communication. Congress still protects about $180 million for polio work, and the Gates Foundation has pledged $1.2 billion for 2026 to 2029. Eradication remains within reach only if countries continue to treat it as an essential public health priority. 4.2. When should you treat a fever? Evolution holds the answer. Evolution shapes interventions from antimicrobial resistance and vaccination to cancer therapy, agriculture, and law. Antibiotic use selects resistant microbes, and targeted cancer treatment can remove one cell population while releasing its competitors. Adaptive therapy seeks to kill only enough tumor cells to stop growth while retaining some treatment-sensitive cells, limiting the expansion of resistant clones and allowing patients to live longer with cancer. Fever shows why evolutionary reasoning cannot be reduced to a universal rule. Raised temperature is a natural defense against infection, but many pathogens have also evolved to grow under febrile conditions. A better approach is to identify which viruses or bacteria can exploit fever. If genomic markers indicate that a pathogen continues to grow at high temperature, drugs should reduce the fever. Otherwise, the response might be allowed to run its course. Natural selection can improve medicine, agriculture, and policy, but different evolutionary frameworks ask different questions. Useful intervention therefore depends on the specific relationship among host, pathogen, treatment, and environment. 4.3. Personalized gene therapy relieves severe epilepsy. A personalized therapy reduced seizures and improved developmental abilities in 2 boys with severe neurological disorders. Developmental and epileptic encephalopathies can cause uncontrolled seizures and movement problems, and one common cause is a mutation in a gene controlling sodium flow into neurons. Standard epilepsy medicines are often ineffective. Researchers designed an allele-specific treatment for each child, switching off the disease-causing copy of the gene while leaving the healthy copy intact. One patient was 9 years old and the other 14, and the therapy was tailored to each genetic variant. Seizure frequency fell by 26% in the younger boy and by 90% in the older boy. After treatment, the younger child stopped a drug he had taken since birth, and the older child became able to walk independently. Neither experienced an adverse side effect during the reported follow-up, but longer observation is still required to establish safety.
4.4. Mutation-dependent responses to sleep and exercise in clonal haematopoiesis. Clonal haematopoiesis activates inflammation and raises the risk of atherosclerosis. As blood-forming stem cells age, they acquire somatic mutations, some of which give a clone a survival or growth advantage. In atherosclerosis-prone mice, uninterrupted sleep or exercise suppressed expansion of JAK2 V617F and TET2 loss-of-function clones, but did not suppress TRP53 loss-of-function or DNMT3A R878 clones. Sleep and exercise also reduced atherosclerosis driven by JAK2 V617F, TET2 loss, and TRP53 loss, but not disease driven by DNMT3A R878H. The interventions acted through distinct neural and immune pathways. Sleep and physical activity can therefore support cardiovascular health, but the protection is not uniform. It depends on the mutation that drives the hematopoietic clone. 4.5. The heart has a little brain that protects against stress. Every heartbeat is regulated not only by the brain, but also by the intrinsic cardiac nervous system embedded in the fat surrounding the heart. Researchers identified 2 neuronal populations in mice. Stimulating NPY-positive neurons slowed the heart, whereas destroying them caused heart failure and death, showing that they both restrain excessive rate and help to maintain basic pumping. DDAO1 positive neurons seemed unimportant at rest, but 2/3 of mice lacking them died during blood pressure measurement or other stressful procedures after a sudden, unrecovered fall in heart rate. Stimulating these neurons improved survival under stress. The cardiac neural network is therefore not a passive relay of commands from the brain. It actively protects the heart during acute challenge. Marker genes for the two populations are also expressed in human cardiac neurons, although translation to treatment remains uncertain. The findings could help to refine imprecise procedures such as atrial ablation and might support more targeted non-pharmacological therapies or drugs. 4.6. Rare contagious cancer discovered in wild catfish. Researchers have discovered a contagious cancer in catfish, only the fourth known type in which tumor cells spread naturally from one animal to another. Anglers and wildlife biologists first noticed black skin tumors in brown bullhead catfish from Lake Memphremagog in 2012, and later surveys suggested that roughly 1/3 of the fish were affected. The melanoma can spread to the brain, liver, and other organs, but its overall effect on survival is unclear because tumors occur in young and old animals without an obvious age-related collapse. Genome sequencing showed that tumors from different fish shared hundreds of thousands of variants and formed one lineage distinct from the healthy cells of their hosts. A 1925 paper had described a similar disease in Massachusetts and showed that it could cast a healthy fish. The discovery suggests that transmissible cancers might be less rare than assumed, and that addition.
Section 5. Molecular Biology, Biochemistry, and Genomics. 5.1. Metabolite glues as a means of purine sensing and chemotherapeutic response. Molecular glues stabilize weak interactions between proteins and give the resulting complex a new function. The study shows that natural purine metabolites can perform this role. Adenosine monophosphate and guanosine monophosphate connect the rate-limiting purine biosynthesis enzyme PPAT to the inhibitory protein NUDT5, allowing cells to sense purine abundance and impose feedback control. When purines are scarce, fewer glue molecules are available and PPAT remains active. When purines are abundant, nucleotides stabilize the inhibitory complex and limit further synthesis. The chemotherapy metabolite 6-thioadenosine monophosphate inhibited PPAT roughly 3 times more strongly than the natural molecules and used the same glue interface. Metabolites are therefore not only substrates and signals. They can physically organize protein complexes and alter cell behavior. Thousands of cellular metabolites still have incompletely understood functions, so additional endogenous molecular glues and possible therapeutic uses might remain undiscovered. 5.2. 10 years of mapping gene expression in tissues. The outcome of a gene expression program depends on where a cell sits within a tissue. Researchers therefore sought a method that could analyze all RNA transcripts while preserving microscopic spatial information, showing both which genes were active and where they were active. The effort began around 2009 and required a decade of curiosity, repeated failure, persistence, and funding able to tolerate long timelines and uncertain commercial return. Spatial transcriptomics eventually made it possible to map molecular activity across intact tissue. 10 years later, scientists are still beginning to understand what such maps can reveal about development, disease, and cellular interaction. Long-term public support in academic laboratories created the foundation, while industrial investment increased scale, accessibility, and the number of researchers able to use the technology. The continuing feedback between academic discovery and commercial development is expanding what spatial gene expression mapping can show. 5.3. Cellular water potential sensing through biomolecular condensation. Water is the solvent of biomolecules and is indispensable to cells. Yet the way a cell directly senses a fall in water potential has remained unclear. Researchers identified a sterile alpha motif protein called SAM8. In a screen, SAM8 formed nuclear condensates after heavy water treatment, whereas other known phase-separating proteins did not show the same response. When plants were transferred for 5 minutes into isotonic buffer containing more than 40% heavy water, SAM8 rapidly assembled into visible nuclear condensates. Under osmotic stress, normal plants retained messenger RNA in the nucleus, but SAM8 deficient plants did not show the same response. Biomolecular condensation can therefore act as a direct sensor of cellular water state, converting a physical change in water potential into altered gene expression. The mechanism provides a route by which plant cells can adjust to drought and high osmotic conditions. 5.4. Inner light, in search of life's glow. In 2025, researchers placed 4 anesthetized mice in a dark chamber and used a highly sensitive camera cooled to nearly -100 degrees Celsius. The camera recorded an ultra-weak stream of photons from the animal's skin. After the mice died, the mouse dyed glow almost disappeared. Biophotons arise during aerobic metabolism when reactive oxygen intermediates relax into more stable states and release light. So infection, injury, and oxidative stress can alter emission intensity and wavelength. Teams are testing whether these signals can distinguish healthy and cancerous cells, assess organ or seed viability, or reveal signs of neurodegenerative disease. A more controversial possibility is that cells detect the photons and use them to communicate. Experiments with transparent barriers, isolated mitochondria, and temporal patterns in germinating seeds provide hints, but results are difficult to reproduce and often fail to exclude chemical transfer or background light. Researchers have created an open network and low-cost light-proof equipment to standardize methods. Biophotons are a measurable phenomenon, but their value as diagnostics or as a cellular and neural signaling system remains unproven.
5.5. Nucleotides act as molecular glue. A major class of modern therapies works through induced proximity. Instead of simply blocking one protein, a molecule brings 2 partners together and triggers degradation, modification, or activation. Researchers found that natural purine nucleotides can act as such molecular glues. Cryo-electron microscopy showed adenosine monophosphate filling the interface between 2 proteins and stabilizing their interaction. The mechanism also helps to explain how thiopurine medicines work, because they partly suppress formation of purine nucleotides. Heme provides another natural example by promoting degradation of the transcriptional repressor BACH1. Many approved medicines, especially natural products and their analogs, might therefore be unrecognized proximity inducers. Thousands of metabolites inside cells might influence biology not only through chemical reactions and signaling, but also by physically organizing protein complexes. 5.6. The mutational landscape of STING-induced immunity. STING, the stimulator of interferon genes, is an evolutionarily conserved immune signaling protein with central roles in host defense, cancer, aging, and inflammation. Type 1 interferon, inflammatory cytokines, and non-canonical autophagy downstream of STING depend on ligand-induced structural change, protein interaction, and coordinated intracellular trafficking. The sequence elements that determine when STING activates or remains silent, however, were not fully known. Researchers created a massively parallel assay and tested thousands of single amino acid variants, linking sequence changes to cellular phenotypes. The resulting mutational map identified regions that control activation, trafficking, and signaling output. It also distinguished variants that strengthen immune activity from those that weaken it. The study reveals internal rules governing a major innate immune protein and offers a strategy that can be extended to other signaling systems. 5.7. structural basis for chaperone-guided assembly of RNA-induced silencing complex. The RNA-induced silencing complex, built from an argonaute protein and a small RNA, is the central effector of RNA silencing. The study resolved how the HSP90 chaperone system helps argonaute to fold, open, and load its RNA guide. Therapeutic small interfering RNAs often carry 2-O-methyl and 2-fluororibose modifications to improve stability, reduce immune activation, and preserve argonaute loading. Most of the chemical pattern used in the cholesterol-lowering drug inclisiran was compatible with argonaute folding. Replacing the 2-fluoro groups at positions 2, 6, and 14 with 2-O-methyl groups, however, sharply impaired folding and complex assembly. The structures show that chaperones, RNA chemistry, and loading intermediates jointly determine whether a functional silencing complex forms. They provide a basis for designing small interfering RNAs that remain stable while still being accepted correctly by argonaute. 5.8. Structure of the pre-initiation complex explains CMGE biogenesis. When a cell enters S phase, the MCM double hexamer, already loaded on double-stranded DNA, must recruit CDC45, GINs, and DNA polymerase epsilon to form the CMGE replicative helicase. Cryo-electron microscopy revealed how SLD2, SLD3, DPB11, and other firing factors organize these components into a pre-initiation complex and coordinate separation of the double hexamer, opening of the MCM gate, and extrusion of the lagging strand. Without SLD2, these transitions were inefficient. Adenosine triphosphate hydrolysis then drove the two CMGE helicases in opposite directions to establish bidirectional replication forks. The structure connects a licensed but inactive origin to the mature machinery that unwinds DNA. It also suggests that the role of yeast SLD2 in lagging strand extrusion might be conserved in its animal counterpart, RECQL4.
Section 6. Materials, Chemistry, and Engineering. 6.1. Graphene oxide-polydopamine membranes with controlled interlayer spacing. Stacked graphene oxide membranes can filter water, ions, and molecules at high speed, but it has been difficult to compress and tune their interlayer spacing at sub-nanometer scale while preserving stability in water and rapid transport. The researchers combined graphene oxide with polydopamine and adjusted dopamine concentration and assembly time to control the channels continuously between 5.9 and 10.2 angstroms. In the dry state, spacing reached 5.9 angstroms. In water, the membrane distinguished rubidium and potassium ions whose hydrated sizes differ by less than 0.1 angstroms, achieving a separation factor of 5,320. The design joins sub-angstrom structural control with stability, high water flux, and precise ion sieving. It offers a possible route for water-salt separation and for separating chemically similar ions that conventional membranes struggle to distinguish. 6.2. Programming fracture resistance in metamaterials via elastic instabilities. Designing fracture-resistant materials is difficult because toughening mechanisms interact across many scales. Mechanical metamaterials offer programmable architecture, but earlier work mainly described how conventional lattices fail. This study used elastic instability to program fracture itself, linking two failure modes that had often been treated separately. Metals can dissipate energy through plastic deformation, yet in large structures, the inelastic zone can remain small relative to the specimen and brittle fracture can still dominate. By shaping unit cells so that they buckle before catastrophic cracking, the researchers controlled where strain localized and how cracks initiated, turned, and stopped. When the difference between the stress plateau and final strength was small, the inelastic region remained confined near the crack, and fracture energy still depended mainly on material and cell geometry. Elastic instability design can therefore do more than change shape or absorb impact. It provides a general way to steer crack paths and increase resistance to failure. 6.3. C-glycoside synthesis through radical cross-coupling of glycohydrosides. Carbohydrates are among nature's most abundant and structurally diverse biomolecules, with roles in energy storage, molecular recognition, and cell signaling. C-glycosides replace the oxygen of an ordinary glycosidic bond with carbon, making them more resistant to enzymatic hydrolysis and valuable in medicinal chemistry. The researchers developed stable glycohydrosides that can be prepared in one step from unprotected natural sugars and then used in radical cross-coupling. By tuning hydroside structure, solvent, base, temperature, and ligand, they improved yield and anomeric selectivity. The method produced all approved C-aryl glycoside sodium glucose cotransporter 2 inhibitors and also enabled synthesis of natural products such as samracin and miopeltolide-related structures together with pharmacologically useful probes. The approach combines efficiency, selectivity, and scalability, providing a general platform for rapidly exploring carbohydrate-derived medicines, natural products, and chemical tools.
6.4. Make me sway. Floors move with the wind. Engineers can reduce dangerous movement in tall buildings by allowing the upper floors themselves to sway. Wind and earthquakes disturb occupants and can threaten structural stability. Many towers use tuned mass dampers, large weights that move out of phase with the building, but these systems are bulky, difficult to install, and usually tuned to a limited set of frequencies. Inspired by traditional Japanese pagodas, the researchers placed the damping mechanism within the building rather than adding a separate mass. The top floors were connected to the core by springs and energy-absorbing components, allowing them to move sideways out of phase with the main structure and cushion its motion. A scale model of a 300-meter tower was tested in a wind tunnel. Its largest accelerations during shaking were about 70% lower than those of a model without the system. 6.5. Ammonia pressure controls colloidal metal nitride synthesis in molten salts. Metal nitrides are important in optoelectronics, energy, and medicine. But colloidal nanocrystals of refractory early transition metal nitrides are difficult to make because ordinary organic solvents and surface ligands cannot withstand the required temperatures. The study used molten salts and controlled ammonia pressure to stabilize nanocrystal surfaces during synthesis. Reactions were carried out between 425 and 600 degrees Celsius under 1 to 5 megapascals of ammonia. Higher ammonia pressure suppressed aggregation and sintering, allowing colloidal particles to remain dispersed. The method produced several metal nitrides and extended colloidal synthesis towards materials with strong covalent bonding. Gallium nitride family semiconductors could make colloidal nitride quantum dots useful in optoelectronic devices, and related refractory compounds might become accessible through the same pressure-controlled approach. 6.6. Stereo-retentive decarbonylative C-C cross-coupling. Cross-coupling methods that form C-C bonds are increasingly common, but retaining the original stereochemistry of a chiral starting material remains difficult. Drug discovery increasingly seeks molecules with greater three-dimensionality and a higher fraction of C-carbon. Existing enantioselective approaches often use chiroligands to control racemic radicals, while organometallic reagents can be limited in availability and functional group compatibility. The researchers converted chiral carboxylic acids into acyl metal intermediates and then used a metal Curtius rearrangement to generate configurationally retained alkyl metal species for coupling with another C fragment. The method worked with substrates derived from amino acids and alpha-hydroxy acids and produced diastereomers that radical stereocontrol would struggle to access. It provides a direct route to stereoretentive CC bond formation and a mechanistic foundation for further stereospecific cross-coupling reactions.
Section 7. Neuroscience, Behavior, and Social Systems. 7.1. Restoring cortical disinhibition improves Huntington's disease phenotypes. Huntington's disease is a severe movement disorder with no cure. It is caused by expansion of CAG repeats in the first exon of the Huntington gene, but the downstream circuit changes that produce symptoms remain complex. Researchers used long-term in vivo 2-photon calcium imaging to follow 3 classes of cortical inhibitory neuron and excitatory corticostriatal projection neurons as disease progressed in R62 mice. Related changes were also found in the ZQ175DN knock-in model. Somatostatin and parvobumin interneurons did not change in the same way, showing that the disease does not simply reduce activity across the cortex. It selectively disrupts particular inhibitory and disinhibitory nodes. Optogenetic activation of vasoactive intestinal peptide interneurons in motor cortex restored cortical disinhibition and moved motor-related excitatory output towards the healthy pattern. The intervention also improved behavioral phenotypes in the mice. Whether the same circuit can be restored safely and durably in people remains to be established. 7.2. Cooperation conflicts with equality when allocating public goods. Cooperation is usually treated as the desirable outcome of a social dilemma, but the way a public good is distributed can change both behavior and prosperity. In the experiment, each person received the same initial endowment and could choose whether to contribute. Under equal distribution, everyone received the same share of the public return. Under proportional distribution, people with greater potential contribution received more. Proportional allocation could raise the relative return of marginal participants and increase total public goods, yet it made it harder for cooperation to spread than equal sharing did. A society can therefore become more productive without ensuring that everyone benefits equally. The problem of promoting cooperation is incomplete unless distribution is considered at the same time. Policy for public goods must weigh aggregate gains against the inequality that a contribution-based rule can create.
7.3. Astrocyte glucocorticoid receptor signaling restricts neuronal plasticity. Sensory experience refines neural circuits during critical periods after birth, but the environmental signals that close developmental plasticity have been unclear. Researchers combined paired single-cell transcriptomics with chromatin accessibility sequencing to follow experience-dependent maturation in mouse primary visual cortex from postnatal day 7 to day 35. Light exposure recruited glucocorticoid receptors to chromatin in an astrocyte-specific manner and promoted astrocyte maturation. Disrupting this signaling prolonged neuronal plasticity and altered the normal developmental program. The work identifies astrocytes as active regulators of the transition from a highly plastic juvenile cortex to a more stable adult state. Disturbance of this pathway could also increase susceptibility to neuropsychiatric disease. 7.4. Sparse-to-dense coding transformation between hippocampal areas CA3 and CA1. The hippocampus is central to spatial memory and navigation. CA3 and CA1 form consecutive processing stages with very different anatomical connectivity, yet their place cell codes look surprisingly similar in small experimental environments. The difference became clear when animals traveled through tunnels 130, 180, and 200 meters long. CA3 represented space sparsely, with fewer active fields, whereas CA1 produced a denser code. Even so, the size of individual place fields remained similar between the regions across 5 environmental scales. The sparse-to-dense transformation from CA3 to CA1 therefore changes the format rather than the basic spatial scale of the representation. It can compress and redistribute spatial information, supporting efficient coding and rapid learning. 7.5. Transcription factor codes patterning neuronal ground plans of the cerebrum. Brain regions that regulate motivated behavior, including the vertebrate hypothalamus and the arthropod central brain, contain specialized circuits for sensory information and internal state. Mating, feeding, and aggression are controlled by many neuron types, each with a distinct computational role. The study analyzed the male courtship circuit of the fruit fly and traced how transcription factors specify neuronal identity. Half-lineage identity established a ground plan. Birth order divided that plan into subtypes, and sex-specific programs added further specialization. These layers of gene regulation formed a nested code. Persistent transcriptional combinations acted like molecular barcodes, allowing one developmental logic to generate circuit elements with different functions and organize a range of motivated behaviors.
Section 8. Research Policy, Careers, and Scientific Culture. 8.1. U.S. agencies push to restrict China collaborations. Lawmakers from both parties in the United States House of Representatives expressed concern that China could exploit American research, but they disagreed over how to protect science and how to fund the institutions responsible for security. At the 15th of July hearing, the head of the National Science Foundation's Research Security Office said that her team had fallen from 10 staff members in 2024 to 5. The foundation had announced a ban on collaboration between senior personnel on its grants and institutions designated as national security threats, including leading Chinese universities. A previous proposal, the SAFE Research Act, would have denied federal funds to American researchers who had collaborated with restricted Chinese entities during the preceding 5 years, but it failed. Analysis of the Dimensions database found that papers and preprints with both Chinese and American authors, and funding from the Department of Energy, the National Institutes of Health, or the National Science Foundation fell by about 40% from their 2020 peak to 2025. The hearing exposed continuing disagreement between broad restrictions, open exchange, and the damage caused by cutting the very agencies expected to protect research. 8.2. Meet the people getting practical PhDs in China. Most doctoral students are required to submit a written thesis. A Chinese law passed in 2024 now allows some engineering candidates to graduate with an innovative product, technique, or project instead. Wu Shangyong, trained jointly by CRRC Qingdao Xifang and Southwest Jiaotong University, developed a hardware and software system that plans, monitors, and adjusts rail vehicle production in real time, then tested it on an operating factory line. Kui Guangzhang created Yansheng, a computing system for training, fine-tuning, and deploying artificial intelligence programs. His product appraisal report ran to 180 pages, and a company he co-founded has licensed the system and earned 25 million yuan. Jia Lin worked with Qingdao High Image Technologies and Xidian University on a chip for artificial intelligence enhancement of television images. It entered mass production in 2023 and is used in Hisense and Toshiba sets. The practical route does not remove research, evidence, or written explanation. It shifts the center of the doctorate towards systems that must work under real production, cost, and market conditions.
8.3. Values in science go beyond politics. Scientists should reflect on their political assumptions, but treating values in science only as political bias makes the problem too narrow. Values enter research whenever people decide what is worth investigating, what counts as relevant evidence, how uncertainty should be handled, which models are adequate for a purpose, and when a claim is sufficiently supported to be communicated as knowledge. These judgments can reflect epistemic values such as simplicity and explanatory power, but they can also involve ethical, social, institutional, and economic considerations. The values that shape inquiry are not always the same as those that shape the use of scientific knowledge. Science becomes more trustworthy when these judgments are made visible and open to criticism, especially when evidence is translated into action. 8.4. A fever dream of jellies and slugs. Entering the world of the naturalist Ernst Haeckel is like falling into a kaleidoscope of vivid sea slugs, coiling octopuses, looping larvae, and bulbous bodies. The art and science of Ernst Haeckel gathers 450 drawings, watercolors, and sketches of jellyfish, radiolarians, nudibranchs, and siphonophores. Haeckel could look through a microscope with one eye while drawing with the other, identified thousands of species, and used his images to promote Darwinian evolution. He also constructed racial hierarchies, endorsed eugenic ideas, and praised the killing of weak or disabled infants, views that must be condemned and were later admired by the Nazis. The book therefore preserves both extraordinary scientific artistry and an abhorrent ideology. Even so, Haeckel's insistence that small and neglected marine organisms deserved aesthetic attention as well as scientific study remains visible in the work. 8.5. Ancient bird carvings found in a German cave of wonders. Hohlefels Cave in southwestern Germany has produced a remarkable sequence of early modern human artworks, including the 40,000-year-old Venus of Hohlefels and some of the earliest known musical instruments. The 2025 field season added 2 tiny bird figures carved from mammoth ivory, one apparently resting and the other with wings spread, together with fragments of a vulture bone flute. The chamber near the cave entrance has also yielded carvings of waterfowl, a horse, an otter, and a small lion man, as well as ivory beads and decorated animal teeth. The objects show highly developed craftsmanship and symbolic expression among Aurignacian people, although their purpose remains uncertain and might have involved memory, group identity, storytelling, or ritual. Researchers are extracting ancient DNA from sediment to trace the origins and descendants of the earliest occupants, and to reconstruct how the cave was used. Hohlefels is unlikely to be the beginning of art. It is an important point in a wider expansion of people who arrived with a developed artistic repertoire.
All right, we are now ready to enter today's main program. Before we do, please like, save, and share this episode, so that other people who need it can also see today's content and become better versions of themselves. Thank you. You can now use the timeline in the video description or the pinned comment to find the articles that interest you and listen to them in depth.